Welcome to KMR Advice. Today, we are diving into the intricate world of bronchial asthma. In this presentation, we will explore the physiological mechanisms underlying asthma, including the roles of the sympathetic and parasympathetic nervous systems, as well as the impact of adrenal medulla secretions like adrenaline. We'll also discuss the involvement of non-adrenergic, non-cholinergic mediators and various autocoids in bronchospasm. Finally, we'll delve into the pathogenesis of asthma, examining both the immediate and late responses that contribute to this complex condition. Stay tuned as we unravel the details of asthma pathophysiology. Welcome to our presentation on the physiology of bronchial asthma. We'll explore how the sympathetic and parasympathetic nervous systems affect the airways. The sympathetic system, through beta-2 receptors, causes bronchodilation and decreases secretion via alpha-1 receptors. Conversely, the parasympathetic system leads to bronchospasm and increased secretion through M2 and M3 receptors. Role of adrenal medulla and NANC mediators. The adrenal medulla releases adrenaline, which acts on alpha-1 and beta-2 receptors, leading to decongestion and bronchodilation. Additionally, non-adrenergic non-cholinergic, or NANC, mediators, such as neuropeptides, often cause bronchospasm and play a role in the pathogenesis of bronchial asthma. Autocoids and bronchospasm. Most autocoids, except for vasoactive intestinal peptide, or VIP, and prostaglandin E2, or PGE2, can induce bronchospasm, further complicating asthma's physiological landscape. Physiological, clinical, and pathological aspects. Physiologically, asthma is characterized by widespread, reversible narrowing of the bronchial airways and increased bronchial hyperreactivity to inhaled stimuli. Clinically, it presents as recurrent bouts of coughing, shortness of breath, chest tightness, and wheezing. Pathologically, it involves lymphocytic and eosinophilic infiltration. Changes during an asthma attack. During an asthma attack, the amount of mucus in the lungs increases and becomes thick and sticky. The cells lining the bronchial tubes become inflamed, causing the tubes to swell, and the muscles around the tubes contract, leading to constricted hypertrophic muscle and inflamed cells lining the airways. Triggers of asthmatic symptoms. Asthma symptoms can be triggered by allergens such as pollen, house dust, mites, animal dander, molds, foods, and insects. Other triggers include strong odors, exercise, cold air, pollution, emotional stress, drugs like aspirin, beta blockers, ACE inhibitors, and upper respiratory infections. Pathogenesis of asthma, immediate response. The immediate response in asthma pathogenesis involves the release of histamine and tryptase, leading to bronchoconstriction and inflammation. In asthma, the immediate response is triggered by exposure to allergens or irritants. This phase starts with the activation of mast cells and basophils in the airways. Upon activation, These cells release various mediators, including histamine and tryptase. Histamine is a key player in this phase. It binds to receptors on the smooth muscle cells of the bronchi, leading to bronchoconstriction, which results in narrowing of the airways. Histamine also increases vascular permeability, causing fluid to leak into the tissues and leading to edema or swelling of the airway walls. Tryptase, a protease released by mast cells, contributes to airway remodeling by activating other inflammatory cells and degrading extracellular matrix components, further exacerbating airway narrowing. The immediate response is characterized by acute bronchospasm, increased mucus production, and airway inflammation. These changes lead to the classic symptoms of asthma, such as wheezing, coughing, and shortness of breath. This phase occurs within minutes of exposure and can last for several hours. In the late phase of asthma, inflammation persists, leading to further immune system activation and contributing to chronic symptoms, and airway remodeling. The late response in asthma pathogenesis occurs several hours after the initial exposure, and can last for days. This phase is driven by the sustained activation of inflammatory cells, particularly eosinophils, T lymphocytes, and macrophages, which have migrated to the airways. Eosinophils play a central role in the late response. They release cytotoxic granules containing major basic protein, and eosinophil cationic protein, which damage the epithelial cells lining the airways. This damage contributes to persistent inflammation, and hyperreactivity of the airways. T lymphocytes release cytokines such as interleukin-5, IL-5, and interleukin-13, IL-13, which further promote eosinophil activation and mucus production. These cytokines also contribute to airway remodeling by stimulating fibroblast proliferation, and collagen deposition, leading to thickening of the airway walls. Macrophages release additional inflammatory mediators that perpetuate the cycle of inflammation, and tissue damage. The late response is characterized by chronic inflammation, airway hyperresponsiveness, and structural changes known as airway remodeling. These changes can lead to persistent symptoms and reduced lung function over time. Effective management of the late response is crucial to prevent long-term complications and improve asthma control.